Hello. Thank you everyone for joining us. I know this is a busy time of December. We were actually just chatting about how crazy these first 15 days are as, universities and students are working through their timelines. We're also talking about Christmas. How is that possible? My name is Katie Hall, and we want to welcome you to our Q4 webinar on tailoring soy to catfish. We're excited to discuss the benefits and opportunities we see for soy-based feeds to make an impact that supports U.S. aquaculture and U.S. soybean farmers. Before we get started, my name is Katie. I serve as the executive director of the Soy Aquaculture Alliance, and I want to start with a few housekeeping things. So first and foremost, this webinar is being recorded, so I'll be available to reference. So it'll be available to reference later. I'm sorry. It'll also be on our website and distributed through our newsletter. If you need it and you can't find it, don't hesitate to reach out to me too. I'd be happy to send it to you. Number two, please don't be shy about asking questions. Um, it's always fun to have an engaged audience, and these guys are ready for whatever you might throw at them. Speaking for you guys, but I know they've got it handled. So we'd love to hear from you. And last on the housekeeping list, this wouldn't be possible without U.S. soybean farmers and the U.S. soybean checkoff. We have funding for research, the Soy Aquaculture Alliance, and this webinar because of the soybean checkoff. So thank you to U.S. Soy and our 12 state soybean associations. Thank you to our partners at the U.S. Soybean Board, the World Initiative for Soy and Human Health, also called WISH, and the U.S. Soybean Export Council and American Soybean Association. As we kick things off, hello and thank you to Dr. Davis and Dr. Bruce for taking the time to work through the details with us on today's webinar. How are you each doing today?
We're doing great, Katie.
Great, thank you.
All right, so today we'd love to share some information with you. We very much appreciate the opportunity to do this. Of course, the funding from our soy farmers and Soy Aquaculture Alliance. My name is Alan Davis. I'm with Auburn University, and I am the resident nutritionist, but I also do a lot of work with production and stocking density. I'll be co-piloting this presentation with Dr. Timothy Bruce. I'll let him do a quick intro.
Yep, thanks, Alan. My name's Tim Bruce. I've been at Auburn now, coming up on about 5 years. Here working a lot with fish health, disease work, and then also some overlap into nutrition. So Alan and I get a chance to collaborate quite a bit. Thank you.
All right. So again, we love to share some work that we've been doing with catfish and just give you a little bit of background in terms of the industry. And it's a thriving industry that's been around quite some time. It is one of the largest aquaculture segments here within the United States, primarily in Alabama, Arkansas, and Mississippi. Now, there's a rich history and evolution of the catfish industry from the 1950s on through today, and a lot of neat things have been done and developed through that catfish industry to make it the industry that it is today. Now, again, since it has a long history, it is what I would consider a very mature industry. It is very well developed in terms of the components of that industry are there. Unlike a lot of our other species where all these components are not there, but from the egg all the way to the plate, we have well-established processes and players who are producing the fish, who are processing the fish, producing the feeds, and of course, taking that out to the final market. Now, within the U.S., there's about 400 catfish food farms, again, in Mississippi, Alabama, Arkansas, Texas, as well as Georgia and a few other places, but 3 to 4 primary states doing the production there. And it is a very important industry to our aquaculture system. It has an impact to local economies of about $2 billion. And it's about 75% of the aquaculture produced within the United States. So it is an important industry. And this food fish does compete with other protein sources, right? So our catfish is competing with other fish, but also other proteins such as poultry or other meat products. Now, the industry is very similar to other aquaculture industries. On the right-hand side, we've got a list, one of the surveys looking at problems within the industry. And on the left-hand side, we've got some example of problems that occur in the catfish industry. And they're very similar. But one of the things that is often near the top of these lists for almost all the industries is the need to improve the feeds and have those feeds as an inexpensive component. Of course, survivability, profitability, problems that occurs also are on those lists. Now, nutrition, in terms of catfish nutrition, historically, this has been reasonably well funded. I'm not going to say we couldn't use more funding. We don't have all the answers, but it is one of the better studied fish that's in aquaculture. Okay, we got a lot of different species in aquaculture, but there's quite a bit of research that has looked at the nutritional requirements of this species, looked at the different ingredients that can be used, how to formulate feeds, and we're really keying in a lot of the times on reducing the cost of the feed used in the industry. So a lot of work has been gone into that. But the industry, like all other industries, is under a lot of competition, right? We have to increase the efficiencies if we want to remain viable as a commercial industry. And so, of course, one of the ways to do that is to continue to improve the feed, continue to look at different ingredients, refine our nutrient requirements to do that. Another way to do that is simply to shorten the time to market. And actually, this works across many animal production systems. The shorter that we have an animal in our hands under production before it goes out to the market, the less time it has to have problems. It's very simple. If I can have a faster growing animal or reach market sooner, there's less likely that I'm going to have a problem. I'm going to run into diseases or predation or whatever might cause problems in a farming situation. So again, also producing these fish in a faster pace is very important, which oftentimes means improved nutrition. So if you look at our industry, as I said, it's very mature. We use bulk feed that's taken out to the farm. You can see a bulk feed truck loading a bulk bin that is loaded out in bulk to our feeds that then distribute that feed out to our ponds where the fish are. The development of commercial feeds, floating fish feeds, and the management of them is actually quite good in the catfish industry. If you look at a generic feed, and again, this is just kind of an example of what a catfish feed might look like. It's going to be like any other feed. We have trace nutrients that we have to provide to the animal, so vitamins and minerals. Maybe some dietary supplements for health or for enzyme activity might be added. The biggest component of that feed, of course, is going to be the carbohydrates or the energy source, as well as the protein source. And so if you look at the carbohydrates that are there, that would be like corn or rice. In this example, we have corn about 20% of the diet and wheat middlings about 24%. So these are the carbohydrates that actually we need for an energy source for our animals. But also it allows that feed to puff up and make a floating feed, which is required for us to be able to observe the consumption of that food. The other big component of that feed, of course, is the protein source. And if we look at the proteins in catfish, we can actually take out all the animal protein, but it's more cost effective, more efficient cost-wise to have about 8% of an animal protein. The rest of that protein is coming from plant-based ingredients. And, as small component, we might have, in this example, cottonseed meal or ethanol byproduct meals or some plant-based ingredient, but the vast majority of the protein coming into these feed formulations is coming from solvent-extracted soybean meal. And it makes up a good portion of that feed, and it's a very important component in terms of nutrition and delivering protein to our animals. Now, I look at the use of plant-based animals. I work with a number of species, so I have to think about a range of species. And the way I think about the use of plant-based feed formulations is we have some animals that are extremely tolerant. So you see them here at the top. We have some animals that are not so tolerant, okay? For example, our salmonids, they actually have some allergic responses to some of the plant-based ingredients or anti-nutrients that are present there. And so we have to be careful with the quantities of soy that are used in those formulations or the type of soy that is used. Other animals, such as a lot of our marine species, say the Florida pompano, the California yellowtail, white sea bass, they need some animal protein, but they're very tolerant of plant-based proteins. And then you get to, for example, our species we're talking about today, catfish, they're extremely tolerant to these ingredients, and they actually tolerate a very high level in their formulations. Now, the problem that we run into as we move from an animal-based feed to a plant-based feed, which is most of what we're using in our aquaculture diets these days because of cost and sustainability, is that those plant-based ingredients bring with them some anti-nutrients, some fiber. There's lots of different ways to look at it. But if we talk about just the fiber component, that's not found in other ingredients. It's found in those plant-based ingredients, and those can potentially reduce the performance of the animal. So we need to be aware of them and be able to look at what happens with them. Now, again, soy is one of the biggest protein sources in the world. It's also one of the primary protein sources in all animal production. That's the reason we do a lot of work with it in terms of optimizing that. But soy, especially that's produced here in the US, as well as our corn, our rice, our other agriculture products, are all very sustainable. And that means that's good for us to be using them as primary ingredients and primary protein sources. Now, there's lots of advantages to soy, but there are also potentially some disadvantages. Now, as a primary protein source, it has very good amino acid profile. It has a relatively low level of anti-nutrients. It's highly digestible, and it's available throughout the world. That's the reason it's used in a lot of feed formulations. And it has a pretty favorable price and availability. Consequently, it's high use in feed formulations.
Yep. So with that, and just to take over, reiterate what Alan said, we have an optimal protein source here for inclusion in a lot of our catfish diets. We see a lot of great responses to that. Again, there become some factors that we really have to monitor as we go throughout really choosing that inclusion rate for them. So with catfish as a whole, coming from the background more of disease work, as Alan mentioned, that's a big issue. Bacterial diseases, it really goes hand in hand with enteritis, issues caused from the anti-nutritional factors, and the move in of the gram-negative bacteria to really cause septicemia or dysbiosis in the gut. As Alan also mentioned, what I've really noticed from my time here at Auburn working with catfish production is the fact that that is really your time to interact with those fish to get a good grasp of how they're feeding. And also that in pond culture, that's a crucial moment if you want to actually provide insights and interventions like vaccination, for example, or therapeutants. It goes hand in hand with that. So feeding really is a cornerstone to that. Again, going back to those anti-nutritional factors, a lot of that's going to come down to the processing methodologies employed, as well as the genetics or strain selection on top of them. So to list some, we're familiar with quite a few. We've got, of course, trypsin inhibitors, proteases, lectins, phytic acids, a lot of things that can be mitigated through processing, but what overall is the impact on those ANFs across different species, going back to Alan's scale? Well, this is an example here of distal intestine in some largemouth bass that we just finished up some SAA work. And what we often see is sort of a breakdown of efficiency and structural issues with the distal intestine when we get soybean-based enteritis in those fish. So we're going to start seeing those ANFs play a role with digestibility and then cause some pathology related to the villi, absorption capabilities, and overall immune cell infiltration into those areas. So it's something that we really need to take into consideration if we're trying to maximize the amount of digestibility that we get out of our products. And with that, as I had mentioned, a few of the avenues are providing processing. That may be the addition of different chemicals or enzymes to those steps, maybe biological reagents added in, genetics, selecting, as we'll talk about, for low oligosaccharide soybean meal. The oligosaccharides cause issues with sugars that can serve as ANFs, and they cause issues with dysbiosis in the gut, as well as undue fermentation processes that allow for decreased digestibility and nutrient uptake. So with that, what we're hoping to do through these main methodologies, lower the fiber content, enhance the protein profiles, and of course, lower ANFs to make them tolerable and optimize performance for the species that we're working with.
All right, so back to our study that we're working on now. This is a combination of a number of studies that we did, so it's going to be very data heavy. So I'm going to drop off a little bit of the details. So if anybody does have questions of specifics, drop us an email. So our objective was, because our catfish diets are very soy-centric, we'll just say. They use a heavy amount of solvent-extracted soybean meal, which potentially has, like Tim mentioned, anti-nutrients or high levels of fiber that they're bringing into the feed formulations, which may have adverse effects on the animals. We wanted to look at some of the newer varieties of processing or genetics and see if we could improve these feed formulations, because we would like to be able to, again, increase the growth rate or have better economics in terms of catfish feed formulations. So if you look at our diets, again, our basal ingredient, so our primary protein source, is solvent-extracted soybean meal. We happen to have a Bunge plant not too far from us, so we tend to get all of our solvent-extracted soybean meal from them. That is what is on the commercial market, and that is the majority of the protein that is being used in feed formulations. But then we have these newer products. So for example, our low oligosaccharide soybean meal, which is a genetically improved soybean meal. This was processed the same as solvent-extracted soybean meal. It just is a different variety that has lower levels of oligosaccharides and higher protein levels. We also went out and sourced for enzyme-treated soybean. So again, this is a soybean meal. It's different. It's been gone through an enzymatic process, again, to digest or degrade some of those anti-nutrients, some of those carbohydrates, some of those fibers, again, producing a higher protein meal with lower levels of anti-nutrients. And then lastly, what we wanted to look at, now I do a lot of work with corn-based products, and I do a lot of work with soy. And the reason I work with them together is because they make a nice balance in terms of amino acid profile. So we wanted to bring in a corn-based product, one of the newer ethanol products that's out there, corn fermented protein, bring it into the formulation because it makes a very nice balance of amino acids when it's mixed with soy. There's complementation of the lysine values and the methionine values. In one, it's high, and the other one, it's low, so they complement and make a nice blend. And so these are our four primary ingredients that we're looking at. We have our basal diet, which is based on solvent-extracted soybean meal. Then what we're going to call is our enzyme-treated or ET diet, which is using that enzyme-treated. Our LO diet, which is using the low oligosaccharide. And then a corn-fermented protein-based diet, or CFP as the short terminology. Now, several diets were made for our production so that the diet composition changes a little bit with different batches. So I'm just going to show you one formulation, but this is our solvent-extracted soybean meal, 56% of the diet. So it's primarily solvent-extracted soybean meal. Now we're using some poultry meal as our animal protein source here at 8%. What we simply did is changed out that solvent-extracted soybean meal with our enzyme-treated meal on a protein-to-protein basis. Change it out with our low oligosaccharide, again on a protein-to-protein basis, or reduced by about 10% the solvent-extracted soybean meal and replaced or brought in about 10% of corn fermented protein. Again, keeping the protein levels very similar. But amino acid profiles are very similar across the board, as you can see in this diagram down here. So the approximate composition amino acid profiles is very similar across these test diets. Now, the primary thrust of this research was to do outdoor pond work, okay? But I love to run our indoor systems parallel to our outdoor ponds because in an indoor RAS system, that's where we can get the most resolution or the best resolution. So we're going to show you data from our indoor system, as you can see here in our RAS system at our open wet laboratory. We're going to share you our data in our outdoor ponds. We chose to do this in our in-pond raceway systems for a couple different reasons. Number one, we wanted to demonstrate the efficacy of in-pond raceways in terms of a tool in aquaculture. But as a research tool, it's also a very nice tool because we can put all of our treatments within the same pond. And so if we do this in independent ponds, we then have variability due to the natural productivity or the characteristics of each individual pond, as well as the effects of our treatments. Now, for those of you who are not familiar with in-pond raceways, I'll just take a quick second to show you how they work. Basically, in our example, we're using an airlift to move water. So you have something that's going to move water through the raceways. In this case, we have an airlift right here, and it basically aerates the water and lifts the water, creating a current that runs through that raceway. So essentially, with 12 of these raceways in a single pond, we can have our replication and we have very similar water quality for our experimental design, which is very nice from a research standpoint. And of course, we can also raise large animals. Now, all of our experiments, we're going to do water quality and follow water quality, but water quality to me is pretty boring. We maintain it within acceptable ranges. So just going to remind you that we do it. I'm not going to show it for all of the trials that we're going to run through. We also collect, in addition to looking at the feed intake, looking at the final weights, the biomass, the survivals, we're also going to collect blood and tissue samples for all these experiments. And again, we're going to show you a subset of all of this data because we simply don't have the time for everything. But we did collect blood in all of these to look at the health of those animals. In this particular case, we used the vet scan, which gives us a nice profile of that blood, and we can see what's going on in that. And then also we collected tissue samples so that Tim could run his fancy gene expression, do all his molecular work that he wants to do depending on the objects there. So I'm going to go through results. And remember, we got kind of two different sets of results. One is in our RAS systems. So I put a picture of the RAS when we're talking RAS, or I put a picture of the production when we're talking production. So this work is with the channel catfish. And these are the 4 diets that we had commercially produced. In this example, we actually used one of our commercial diets from the industry as a reference. And you can see we got to final weights. These are small animals grown in the laboratory. But in our low oligosaccharide group, we got a little bit larger animal, statistically larger animal, which also gives us good feed conversion. And in general, our commercial diet did a little bit less. So that shows that there's room for improvement in our commercial diets. Now, if you look at this graphically, it's a little easier for me to see things. I like to make tables, but I don't like looking at them. It's real easy to see that this low oligosaccharide diet outperformed the others and also had a better feed conversion, indicating there's some advantage in the channel catfish in juveniles for the low oligosaccharide diet in terms of improving performance. Now, we also looked at blood work. I'll let Tim give a quick explanation of that.
Yep. Thanks, Alan. So one thing we looked at was we drew sera from these fish at the end of the trial and wanted to look at a variety of different panels, parameters on a panel to sort of get an overview. What's liver function looking like? What does electrolyte balance? And from a physiological perspective, that provides us with some insight on the overall health and well-being. So for example, we have albumin at the top. We did some amino acid transporters, bilirubin, phosphorus, sodium, potassium, and have a nice picture of overall what should be going on with the physiology of those catfish at the trial end. We did see some minor elevations with total bilirubin, which is usually from the breakdown of red blood cells. although significant, probably not much of a physiological impact. And then we did see some minor changes with creatinine, but overall, it looks like liver function, electrolyte balance due to associated with osmoregulation, and protein levels, especially I'm interested in the globulins with the immune response. Those all seem to be relatively impact. So blood was one of the things we collected at the end of this trial period. And then, of course, we also took the distal intestine and liver to get a better view of what's going on through targeted gene expression. So here we have the liver expression profiles. We have the basal diet, the treatments that Alan just referenced, and then also a reference control group. We looked at different markers related to G6-phosphate, for example, fatty acid synthase, growth hormone receptor, hexokinase, IGF-1. We did see some minor differences in hexokinase across the board, but overall, when it came down to growth performance and changes in the liver, we did not see much to discuss. Our main target, however, was the intestine. And this is the gene expression from the tank trial at the end. We had 4 replicate tanks here. We looked at amylase, which is an enzyme involved with starch breakdown. Trypsin itself, I mentioned earlier, trypsin inhibitor is an issue with the ANFs in soybean meal. Didn't see any differences there. But what was interesting is we found differences with the CCK receptors, A and B, which are related to digestion, and nutrient absorption processes. They sort of go in an axis through the neural enteric pathways. And we did see the same conserved trend, which was really interesting across both of those receptor types for CCK. So there is an involvement to some degree with sort of appetite, nutrient processing, and uptake with those, which was quite interesting and something for us to look at a little bit further. And next up, we have hybrid catfish. So we're going to jump across species. So with hybrid catfish production, this is a species that is raised in Alabama. However, primarily hybrids are raised in Mississippi. That's been going on for decades now. Hybrid catfish have different growth mechanics, different disease susceptibilities, survival rates, and have sort of an ability to really do well in intensive culture situations. So Mississippi has adopted this over the years with lots of research done. I should mention with hybrid catfish, that is, of course, a female channel catfish crossed with a male blue catfish. So you do get an element of hybrid vigor throughout. And when you are processing these fish too, you have to set up your processing lines differently because the morphology is a bit different across the whole. So with that, we jumped into some hybrid work as well to line that up with the channel work.
So again, with the hybrids, show you some aquarium work using the hybrids. Unfortunately, when we were doing this work, one of the ingredients are low liquors are, one of our diets was not available. So we only have the 3 diets that are run with this particular aquarium trial. But again, we got a little bit difference in terms of protein retention, which is how efficient they're using protein, and the low oligosaccharide did a little bit better. Didn't see a whole lot of differences in terms of weight gain and performance, but again, a little bit difference in terms of protein retention, and then also a little higher intraperitoneal fat, which would indicate that more of those nutrients are being absorbed and they're being used and stored as an energy source in terms of fat. Now, we had an opportunity to actually do some research diets alongside these commercial diets, 4 diets that we're talking mostly about, to look at the use of that low oligosaccharide feed, to look at the enzyme treated, and also look at a soy protein concentrate, which is now being produced here in the United States, which is now available. And so we use very similar trend as what we've been talking about. We took a soy-based diet, 50% soybean meal, and in this case, we did 50% replacement or 100% replacement using each one of those meals that I aforementioned. And this data was done in parallel with the data I showed you on the hybrids with the commercial versions. Again, this was done in the laboratory to see the response. And you can see across the bottom here is our p-values, and there wasn't a whole lot of difference in terms of the performance of the hybrids to either the low oligosaccharide soybean meal, the enzyme-treated soybean, or the soy protein concentrate. All of these products worked very well and gave good results. Now, on to the in-pond raceways. So again, looking at those 4 diets in these in-pond raceways, we did 2 sets of trials, one with fingerling to a advanced fingerling, and then that advanced fingerling was grown out to a commercial size product or commercial size fish. But all the diets in terms of performance, this is our kilograms of performance, and then our weights. You can see there's very little difference, but I have this graphically, so let's just jump to the graphics. You can see there's very little difference in terms of our final weights or in terms of the kilograms in terms of biomass. No statistical differences there. A little bit difference in terms of our feed conversion ratio, with the low oligosaccharide variety having a little bit better performance, again, reducing our feed conversion, although growth rates were not significantly different. Again, we look at these 3 diets or 4 diets at the end of one of these in terms of our hybrids. Because there was no availability of the low oligosaccharide product, we were not able to run that. And so we only have 3 of the diets run in this particular final grow out. But as you can see, got to good biomasses. We got our animals up to commercial size, about 1.25 pounds in terms of a commercial size catfish. And you can see here across our p-values, with the exception of hepatosomatic index, which would be the size of the liver, there's really no differences in terms of performance. We had similar biomasses, similar feed conversions, protein retention values, interperitoneal fat was very similar, but we did have a little bit shift in terms of the size of the livers, as well as a little bit shift in terms of the dress out, with the basal and the enzyme treated producing the better dress outs, with the corn fermented product having a slightly less dress out, which would indicate maybe the protein is not quite as available as it is in the other diets. So again, looking at that graphically, you see that there's very little differences across weight gain, final weights, biomass production, or our feed conversion, although the enzyme treated did produce a little bit lower level of feed conversion. We also looked at the blood, as Tim had talked about earlier, not seeing many differences there. So we'll go ahead and move forward. Now, again, looking at the channel cat or the hybrid catfish, same process in our in-pond raceways, again, doing that early juvenile production and then moving to final grow out. So have 2 production cycles with our animals, our basal diet here, and then enzyme treated diets down the row. I got this graphically, which will have things, but as you can see, we have very little differences in terms of our p-values here across the bottom, with protein retention being a little bit better right here. And again, I think this was our enzyme treated having a little bit better protein retention. I was wrong. It's the low oligosaccharide that had a little bit better. So again, showing in terms of final weights, average weights, in terms of our production from that final grow out, as you can see, we're getting up to about, again, 1.5 pounds, 1.25 to 1.5 pounds in terms of our final weights, going from that advanced fingerling to our final production size. We have good, reasonable feed conversions, good protein retention, no major differences in terms of the performance of those animals. So again, looking at that graphically, again, you can see that that enzyme treated had a little bit better feed conversion in terms of the performance, though, in terms of weights, in terms of kilograms, in terms of survivability, all of them very good with very little differences across the board. Now, across the experiment, again, we took our solvent extracted or traditional soybean meal and looked at these more processed meals, the enzyme treated, the low oligosaccharide, or looking at one of our newer components from our ethanol industry, our corn fermented protein, as a mix with that. And all of those diets did very, very well. What we see across species is that there are very big differences between species. In the case of the channel catfish, which is very tolerant to plant-based ingredients, we saw very little difference. We did see some performance enhancement with both our enzyme treated and our low oligosaccharide, depending on which experiment we're talking about, but there was no major problems or issues. When you move to, say, Florida pompano or bass or other species that are a little more sensitive to these oligosaccharides and anti-nutrients, we see an enhancement of performance, just depending on species. So overall, again, with the catfish, which is very tolerant to soy-based and plant-based ingredients, we saw all of these having very good response with just very little advantage of any of the newer products, but some advantages in terms of feed conversion, depending on which study we're looking at. So again, from my standpoint, a lot of these products make sense in terms of our fingerling production for our catfish because the higher protein content, they fit better in those formulations. But that's up to our feed formulators to look at that data and make those decisions. In terms of the future of the catfish industry, hopefully that's going to continue to be a strong industry here in the U.S. With continued research and development. Hopefully we can maintain that market here in the U.S. and we'll be happy to answer any questions that anybody might have. Open this up for questions.
Yeah, thank you, Dr. Davis and Dr. Bruce. We really appreciate your review today. It's so fascinating. I'm learning a lot about aquaculture, so I appreciate diving into the deep end here. Jumping right into questions from Michael Rust. Thanks, Alan. Nice presentation. Catfish seem very able to deal with ANF that remain in solvent extracted and heated soybean meal. You mentioned that other species like marine fish and salmonids were less able to use standard soy. Do you think using a more sensitive animal, say yellowtail, would have shown more differences among the diets?
So,
and then he
yeah,
says,
great.
never mind. You answered in the last thought.
Actually, yellowtail is pretty tolerant of solvent extracted soy. But the issue is that, every species kind of has a different tolerance to these products. And my oversimplification of this is that solvent extracted is by far the most available and it's the cheapest. And so when that's appropriate, we use that. And then as we're moving towards more nutrient dense diets, particularly the higher protein diets, or we're moving to an animal that's more sensitive, and I can go to the extreme, let's talk about salmonids, then that allows us to move to these other products where it makes more sense. Also, so a lot of these products have better like phosphorus availability. So in terms of pollution loading and other things, there's also considerations there. So there's, a whole series of things that you need to evaluate when you're looking at these products in terms of how they apply, and there's different reasons why they come into formulations. But yeah,
and also to add on to that too, for Mike's question is we did run a lot of these low oligosaccharide variants too in largemouth bass through our separate SAA project. We did histology at the end with their distal intestines and did not find any signs of enteritis as well in those with comparable performance. So it looks like bass, at least, we have some data points that are comparable to the catfish work too.
Fascinating. Another quick question, kind of unrelated to soy, but I think important from a nutritional side of things. For catfish formulation, why cottonseed meal? Could canola meal replace it if more available locally?
Yeah, and we all have kind of our biases towards any ingredient. But there's a range of plant-based ingredients, all that can be worked into, for catfish, they're very tolerant. So yes, canola meal, but it depends. Canola is a generic term, and there's a lot of variability in terms of the strains and whatnot. But canola meal also has some anti-nutrients, so that needs to be looked at. Cottonseed meal is a byproduct that's been used for a long time. Peanut meal, a lot of different ethanol byproducts. Each one of them have advantages and disadvantages, and just depending on the availability as well as the tolerance of the animal and the type of meal, they're going to come into formulations. But yeah, there's a number of different meals that can be worked into these formulations.
That makes sense. There's another question here that I will follow up with Jerry specifically on, but just for lack of, or for the sake of transparency, he says, I'm a fish biologist and past blue catfish farmer utilizing floating in pond raceways from South Central Illinois, having since relocated to Western Washington. I'm reestablishing my raceways in inland still waters to farm trout and steelhead. And his questions are related to SAA research. So, Jerry, I will send you that information on all of the research that we've done so that you have access to it. But it's another call out to the research that's accessible on our website. We have some really great data and information linked there. I know Dr. Bart is listening. He's also part of our research team. He is the key part of our research team, I should say. And so he's done a lot of work to pull some of that information together so that we have a more comprehensive look at what all of the research looks like, especially related to soybean meal and soy-based feed products. And so, you'll see something on that, hopefully soon as well, to see where all of that kind of comes together in a really nice way. Gosh, guys, I think, the one thing I was coming away from this conversation with is like, and based on your studies, I mean, this is a really broad question and really kind of baseline, but I think I'm curious what areas related to catfish and soy diets need more research. Do we need a couple more hours to talk about it?
No, I think that's a great question. Yeah. I guess from my side, related to disease and health, understanding that a lot of these bacterial pathogens, as I mentioned, have a role with the GI tract, understanding the microbiota in the gut at the same time, how that reacts also to different soy products. And then in turn, if you're looking at, probiotic strategies in combination with soy feeds, vaccination at the same time too, I'm really seeing, at least in catfish, that really diet selection and ingredient selection really does make a big deal when it comes down to trying to push forward, health promoting items in those diets.
Yeah, and these new products just need, and new genetic varieties, again, those need to be evaluated in whatever species you want to look at because there's potential for them. But a lot of our species also, we don't have a whole lot of digestibility work, or don't have a lot of work where we've tied processing chains or quality of any given ingredient back to the nutritional value of that ingredient. And that's a lot harder to do in aquatic species. And soybean meal in the US is very consistent, very high quality product. So you're not going to see a huge variation within the United States, but there are variations from north to south, east to west, et cetera, in terms of the quality or the nutrient profile. I shouldn't say quality, nutrient profile changes. So there's always work that can be done in terms of all ingredients.
No doubt. Diving in a little bit more on ingredients. I know you touched on the corn fermented protein. I'm curious why you think that was so successful. How do corn and soy proteins work together in your mind? I know you talked about it being a balance of different amino acids, but just digging a little deeper on that.
Yeah, and I am very soy centric in terms of my feed formulations. I don't deny that. But if you look at it traditionally, going back 100 years, you matched corn and soy, or you matched peas and soy as what the matches were for protein sources. And the reason being is because corn is high in methionine, soy is relatively low in methionine. It's very rich in lysine, whereas corn is relatively low in lysine. So corn products and soy products make a nice match in terms of amino acid profile, or at least that's one reason. And then there's, the products coming out of the ethanol industry have lots of health potential benefits that Tim can talk about more in depth. So there's reasons from a health standpoint or those other standpoints to have mixed ethanol products, but using mixed ingredients always seems to work better than a single ingredient whenever you're doing feed formulations. And so I tend to use a lot of corn soy combinations because of the costs and whatnot.
Yeah.
I think that's a really good call out. There's another comment in here. Inclusion depends on price of these products, right? That is the underlying conversation when we talk about commercial availability, costs, and then logistics on top of it all. I used to work at Cargill, and there's a site in Memphis, and it's a wonderful site, but it's a corn processing site. And there's not a lot of corn grown in Memphis, but it is on, the six class one railroads. Got all of these railroads close to it. So I think the initial thought was, hey, we're in a great location, but not super close to, the bulk of the corn that they were trying to process. So kind of interesting to see how those logistics played out in real time and how that affects that site. Let's see, one more question here. How did the test environment, your raceways versus um, tanks, or I should say open ponds, affect the fish's response to soy diets? And what takeaways should producers know? Just kind of a revamp or a revisit of the overview.
Yeah, and so just realize that, an RAS system, that's just to me the most controlled, easy, the best thing to look at because you get rid of a lot of other factors. And so that's the reason I like to, even if we don't propose to look at something in an RAS system, if we have an opportunity, we will. But the ponds are is closer to what the commercial environment is like, and that's why we're running them in ponds. But you also have between pond variability. So again, because we wanted to demonstrate the use of the in-pond raceways and it allows us to get rid of that in-pond variability, that was one of the reasons we chose to do that in the in-pond systems. And then in terms of, as again, overall from the research, we do see that, these more processed or more genetically improved products, that they do have some benefit, but it's species specific. And in like was mentioned, you also have to consider cost, nutrient delivery. In a low protein diet, it's a much different discussion than if it's in a high protein diet. Okay. So there's lots of considerations, but all of these products work very well in terms of the catfish, and we've seen them work very well in other species as well. Tim, you might.
Yep, I agree. I think the same thing when you have that natural productivity from pond studies inside. We've been doing that lately with a lot of our work too, where we're trying to do diet studies, if not in the ponds for some of the reasons that Alan mentioned, but we're trying to push pond water through tank systems at least to capture as much as we can of that difference in environment and water quality that way too. That's really important for I think especially the microbial communities that are involved with in the intestinal tracts as well as pathogen exposure too. It changes things up quite a bit.
Awesome. I lied. One final question. Are there other untapped seafood species Americans would eat and that thrive on soy-based diets, which more producers should consider raising? This is a wide open question beyond catfish. What do you think? Maybe tell me what your favorite type is, man.
I know what Tim's going to say. Tim, what's your thoughts?
Yeah, let's go with bass. They're fun.
Oh, I thought you were going to say he worked with burbot.
Oh, burbot too. Yeah, that's right. Yeah, Dr. Kane's on the call, so absolutely. Yeah.
So, I mean, again, we're just now looking at really whether or not you have enteritis in a lot of these species. I mean, we assume they don't or they do, but a lot of times they've not been looked at. But we have a lot of species in the U.S. that are interesting from an aquaculture standpoint. But I think the problem is the marketing standpoint and what can you market and what can you get nice dollars for. The bass is very interesting because it's a high dollar product from a sports standpoint and fairly high dollar product from a sales standpoint. But we work with a number of species that come through the lab that are all very interesting. We got a number of snapper out there on the coast. We've got the yellowtail was mentioned. That's an extremely nice fish, and it also is very tolerant of soy. At least my experience with the California yellowtail. But there are a number of species out there. But for me, it's matching that market and making sure that we can grow them commercially. So, great question. But yeah, I too many favorite species out there.
Curious what you think about jade perch.
You're from the north. What about perch, Tim?
Me?
Yeah. Well, I mean, yellow perch have a lot of potential in the Midwest. There's no question there. They're a solid rock species. I can't comment on jade perch though, just yellows.
Cool. All right. Well, truly, thank you, Dr. Davis and Dr. Bruce. I really appreciate your time. My apologies again for the gibberish, and I don't really know what happened there, but we'll get it fixed and get folks those corrected slides. Just a quick reminder, this recording and webinar will be available on our website. It'll be a couple of weeks' time, but you'll also see that come through in the newsletter and then through email. So if you're interested in taking a more in-depth look at the studies, please be sure to check out our research page. We appreciate each of you listening today. And on behalf of U.S. soybean farmers and the Soy Aquaculture Alliance, thank you so much. Have a great day.